So we have about 12 minutes left in this session. At this point, I want to invite all of our other speakers up to stage so we can go through some of our remaining questions. I believe we've got, I know I certainly have more from our pre-fielded questions, and we've got more in this Q&A box that we can ask people before we wrap it up. So let's see. I think the first one will be for Michael and Troy. This is from Michael Silverman. Wait, no, this is to Ajay from Michael Silverman, my bad. In mammalian cells, it seems that conventional kinesin, KIF5, is responsible for peroxisome transport. Can you say more about the findings of KIF1A and peroxisome transport in zebrafish?
Well, to be honest, the use of the peroxisomes in our very initial experiments has more to do with the combination of fluorescent colors we had so that we would be able to visualize something. So we had done some assays earlier on because we had a red tagged peroxisome and we could see the transport very effectively. Our neuro-D is green, and so we didn't have time to develop the color combinations that were necessary. So the peroxisome focus is really pragmatic based on what tools we had at hand. It's not necessarily something I think is directly connected or to some expectation based on the role of KIF1A. So we hope to actually look at many more things, and then at that point we can go back and say how these different things might have contributed to defects in transport of different types of cargoes, but we're not there yet.
Great. Next question is for Jayne. This is referencing some of the work that you mentioned in Pedro's paper, asking about how we know that organelles are stopping at synapses. For example, did you stain or a postsynaptic marker after live imaging?
We did not do any combo live imaging staining. What we do to determine that it's a presynapse is just look at it over time. So prior to our imaging window, we hunt through, find a field, take a brief image that then we refer back to see if the synaptophysmin or V-glute that we're imaging has stayed in that place for a specific period of time. Then we call it a synapse.
Can you hear me?
Yeah.
Yep, go for it.
Or can I talk?
Yeah, go ahead.
So sorry, maybe this is too detailed for this kind of discussion, but I'm wondering about that question, because what if they're just, vesicles sometimes hang out for a long time.
That is, yeah.
Stationary vesicles don't necessarily make synapses. And so, yeah.
Yeah, no, that's something that we've discussed before, but we have shown in just ICC that these things hang out together. So we know that GDP tubulin and presynaptic markers and postsynaptic markers all hang out together. And there's actually some really interesting data, looking at synapse initiation, showing that prior to a dendrite making contact to an axon, it actually, it somehow finds its way to a spot in the axon where the presynaptic or the synaptic vesicle precursors have paused. So there is kind of this idea that these pause sites are significant for synaptic connectivity and maintenance, but you're absolutely right. Not every paused presynaptic vesicle precursor is a bona fide synapse. But by looking at these, kind of in bulk, we're able to, predict that often when we do see those hang out for a long time, it is a synapse. Thanks.
Next question. It was in the Q&A and then it disappeared. I believe it was for Michael and Troy, but it's sort of a question for everybody who's working with cells. Differentiating iPSC lines is, you mentioned there's these differences in the wild type patient-derived lines versus the R2 or 3S lines. Are there any specific factors that are transported by KIF1A in this process that could be perturbing this differentiation?
Oh, that was my question.
Well, the short answer is I don't know. I would like to think, or I want to hope the answer is no. And I would substantiate it from the following standpoint that I would think that retrograde transport or dynein-based transport would be more important for taking up factors from outside, if it's a growth factor or inhibitor or something, and trafficking it back to the nucleus. Yeah, I don't know. Would someone else like to comment on that?
Could I jump in? So we have built some tools here that may help you resolve that. So we have a DOX-inducible system. So what I'm very curious about is, are patient-derived mutations in the iPS cells giving you the same data as if you took the wild-type cells that have been paired with them, differentiate them fully, and then induce the expression of your mutation in parallel? And so is there a memory to the system, a trajectory change that the development is affecting the results or not? And sort of my history is I was very interested for many, many years in proteases that shed ligands in cells. So there's a lot of research that suggests that if base is being misregulated, that differentiation could easily be affected. And there's probably other proteases in that list that are some semi-ignored too, that are cargo, that are autocrine loop systems. So I would be willing to, we want to share what we built. So if you're interested, we can. This is basically virus constructs. So you can just make your own virus, put them on those cells, and find out pretty quickly.
Thing I wanted to just jump in here with is looking. It relates to the expression of KIF1A, right? So KIF1A is mostly neuronal, and isn't it going to get turned on? It's not going to be like, I'm just thinking that you might not have as big of a problem with differentiation as you might think, because it's not, the mutants aren't present during these like upstream steps before you get to the neurons, right? And actually for my work, since I'm not using the patient-derived iPSCs, we just have a wild type line, that we actually have this kind of streamlined induction system. So it's also DOX inducible, it just induces NGN2. We get neurons within like a week, and then we can grow them out for however long we want. And so, thinking about, for those of us like Ajay and I, who are just inducing ectopic expression of mutant, I was like, well, actually probably be better if we could introduce the mutant sooner. We're talking about like looking at neurite extension and things that, are kind of happening before you have a fully mature network. And I'm thinking like, oh, I should probably, like, as we're talking, this is why this stuff is so great. It's because as we're talking, like, I should probably use like nucleofection to introduce the mutants instead of, typical transfection at like day 21 or something. Because then that would allow me, or maybe do both, really, to see like, well, what's it look like with neurons that have the mutant from the beginning, like right when they're extending out their neurites to, mutants that are already set up more. Yeah, I think that this idea of expression, the levels, when and where you need it, are things that we should all be thinking about.
So in the experiments that I showed, we had used RNA. So actually it's expressed in all the cells in that particular experiment. And yet, as I showed you, that didn't compromise development, at least up to the point where the axon had been made. So in that context, it says at least the level that we were expressing didn't cause a widespread enough problem to compromise some of that development. It was only in the transport. Now, I should say that there was no movement of anything. So I think that we had just really bunged the system up with too much of the stuff. And I don't even know how to interpret the fact that nothing was moving. And I think Julia did an experiment just earlier this week or last week, where she did it at a lower dose. And then again, things were moving even with the mutant version. So we have to carefully do these experiments with different doses, and that in itself will be very informative to see at what point the problems arise, which aspect of transport is affected first. So
In your
this
system,
RNA is kind of a very crude experiment, but it still tells us a lot of useful things.
oh, absolutely. Like, is it pretty easy to like follow up with a Western? You know, if you inject this much mRNA, you get this much. Like, well, I guess, or you might want to do like a.
We just do in situ
Like,
hybridization.
what's the level? Like, how much mutant versus wild type do you have? And
Yeah,
then,
you could do it. Yeah, you could just do in situ hybridization of injected and non-injected fish and compare the expression. That's easy.
Yeah.
All right, I think we've got time for one last question, and I see Rick McKinney just put one in the chat. So I'm confused about the model of kinesin binding. This is for Jayne, but everybody, please feel free to chime in. I'm confused about the model of kinesin binding to GDP versus GTP microtubules. Hirakawa and Cross Labs both showed that kinesin binding expands the lattice to be more like GTP, but is the Holzbaur model opposite?
From my understanding of this literature, and actually Rick would know more than me, so the fact that he's answering this question maybe means we need to think a little more about this. But the kinesin binding expanding, that hasn't been shown specifically for KIP1A. I think that's more the conventional kinesin. I mean, you know, the motor domains are similar but not identical. You know, we haven't done any sort of follow-up structure data. This is all just looking, you know, at microtube, you know, just like looking at its binding with turf. So yeah, I mean, I guess the idea is that once KIP1A hits that, you know, GTP-like state of tubulin, it's more likely to detach. And for particularly what, like, structurally is happening there, we don't know that yet. But that's our model.
Great. Our next session starts in 2 minutes, so I do want to give everybody some time to shuffle back to where we need to be for that. But I would like to extend one more thank you to our speakers, and thank you all for your questions. This has been a great session that sort of turned into a mini discussion, which is an awesome way to handle a Q&A, so I'm pleased with how this evolved. So thank you again. From here, we're going to be exiting this presentation mode, and we're all going to head back to the main room via the button to the left of the stage. So we're just going to head back to where we were for the rest of the day for our final session of the day. So thank you very much, everybody.
Yeah.
Thanks,
Thank you.